Hello students! Today's lab will cover drought and evapotranspiration, two important concepts in agriculture and horticulture. Drought is something that is unfortunately out of our control, and water deficiency has become an increasing threat to agriculture and dryland ecosystems, especially due to climate change. After listening to this lecture, I hope you will be able to define a few of these key terms: transpiration, drought and drought stress, and evapotranspiration. Scheduling irrigations and even fertilizer applications often takes into consideration evapotranspiration, or ET, and growers and extension farm advisors use certain tools to measure ET. We'll look more closely at effects of drought and water stress on plant processes, specifically those involved in productivity and other metabolic processes like photosynthesis. Then, after discussing some negative effects, we'll discuss how this can be utilized, in some cases, in production systems. Something called deficit irrigation is often used to purposefully drought-stress crops with certain benefits. First, some terminology. Remember, transpiration is the movement of water from the leaves of a plant into the atmosphere through the leaf stomatal pores. This is also what drives the passive uptake and movement of water from the soil via roots and the xylem. Evaporation is the loss of water from the soil or media surface. Combine these words and you get evapotranspiration. It sounds like a fancy word, but it's just a portmanteau of transpiration and evaporation. So, by definition, evapotranspiration is the sum of water loss from soil evaporation and plant transpiration. New technology has allowed us to measure ET. CIMMYS, the California Irrigation Management Information System, is comprised of a network of computerized weather stations to measure environmental variables such as solar radiation, air temperature, relative humidity, and wind speed. All of this is combined into an algorithm to compute a daily reference ET, which estimates the potential loss of water through plant transpiration and soil surface evaporation from a control setup of a well-watered grass crop that completely covers the soil surface. Along with other pieces of information such as crop stage, soil moisture, and the weather forecast, growers can use this information to schedule irrigations and sometimes fertilizer applications. This allows for proper timing of water applications to best meet the crop needs. Most Californians are painfully familiar with drought. It occurs all over the world. There are these prolonged periods of low rainfall, which puts crops and ultimately people under great stress. Drought is considered the most significant abiotic stress affecting crop yields in some areas, such as in our Mediterranean-type environment. Professor Jack mentioned one of the most historic droughts in U.S. history, the Dust Bowl of the 1930s, where drought, along with poor land management that led to severe soil erosion, resulted in significant economic damage and social changes. The amount of available water in the plant rhizosphere drops below what is required for sufficient growth and biomass production, and drought stress results when this water loss from the plant exceeds the ability of the roots to absorb water, and, if severe enough, ultimately interferes with normal plant processes. The ability of roots to reach deeper soils where water content is less variable than in shallow soils, particularly under drought conditions, has often been associated with drought tolerance. As mentioned earlier, severe drought stress interferes with several plant processes, including photosynthesis. During drought, plants protect themselves from excessive water loss by closely regulating stomatal opening and closing, compromising stomatal function. This leads to reduced food production for the plant. Regarding respiration, water deficits in the rhizosphere leads to an increased rate of root respiration, leading to an imbalance in the utilization of carbon resources and reduced production of ATP. Decreasing water availability under drought stress also generally results in limited total nutrient uptake, and their diminished tissue concentrations in crop plants. Because stomate function is compromised, decreased water availability also inhibits transpiration, which drives water and mineral nutrient uptake. Although the term plant productivity mostly refers to photosynthesis, I'm using this term more loosely to refer to overall plant growth and success. Under drought stress, plants suffer from impaired germination and poor seedling establishment, which reduces crop growth and yield. Drought stress also leads to the production of reactive oxygen species, which increases peroxidation of membrane lipids and degradation of nucleic acids and structural and functional proteins. Various organelles, including chloroplasts, mitochondria, and peroxisomes, are usually the first target of reactive oxygen species produced under drought stress. The middle photo shows the effect of drought stress on the vegetative growth of rice. Both plants were grown under well-watered conditions up to 20 days following emergence. Then, one pot was submitted to progressively drying soil, or drought stress. You can see clearly the visual effects that this had on the plant. The infographic on the slide describes possible mechanisms of growth reduction under drought stress. Under drought stress conditions, cell elongation is inhibited by reduced turgor pressure. Reduced water uptake results in a decrease in tissue water contents, and as a result, turgor is lost. Drought stress also trims down photoassimilates, which are the biological compounds like glucose, which store energy for use by organisms and are formed by photosynthesis. They're also required for cell division. Impaired mitosis, cell elongation, and cell expansion result in reduced growth altogether. The extent of these effects vary with the plant species, stage of development, and the duration and severity of the drought. Drought stress decreases the photosynthetic rate and disrupts the carbohydrate metabolism and levels of sucrose in leaves. Limited photosynthesis and sucrose accumulation in the leaves may hamper the rate of sucrose export to sink organs and ultimately affect the reproductive development. Apart from limited resources, the reproductive sinks, like flowers, fail to utilize the incoming assimilates properly. Growth rates of roots and shoots, or below-ground and above-ground plant parts respectively, are a reflection of resource availability. With limited water, and therefore reduced photosynthetic activity, some research shows a shift of resources to the roots, another sink organ. In some cases, there can even be a stimulation of root production. Root system size and distribution determines access to water. The ability of roots to grow deeper into soil is not only determined by the genetic makeup of the species, but is also a common response to water deficiency. Despite all the negative effects of drought stress, some crops are intentionally water stressed. This is often called deficit irrigation. In some vineyards, for example, water stress leads to an increase in sugar concentration in wine grapes, changing the chemistry of the berry and affecting the wine. In almond and walnut orchards, controlled deficit irrigation can accelerate hull splitting, allowing the kernels to dry earlier and more completely on the tree rather than on the ground, lessening the possibility of contamination by soil-borne bacteria and resulting in a more timely and uniform harvest operation. Water stress can also be less expensive if you water the crops less often, and can be economical if the impact on yield is minimal. Drought stress can be used in landscaping and gardening too. When moving plants from a greenhouse to an outdoor environment, it is sometimes common to harden off the plants by water stressing them to help them acclimate to the outdoor environment and reduce stress later on. And lastly, as mentioned earlier, it is also used to increase root growth, sometimes used in new plantings.